Hello, everyone, and welcome to our third and final Tour Tuesday, which is a virtual experience exclusively for Extension. We're getting a behind the scenes tour today of the bee labs of Dr. Jamie Strange and Dr. Reed Johnson here at the Waterman Agricultural and Natural Resources Laboratory and also up at CFAES Worcester. Today, we're going to learn a little bit about the research exploring how urban growth impacts native bees, how timing pesticide application could help honeybees, and why honeybees and soybeans make a good pair. So hold on tight. Now, for those of you who are watching live, please remember that we're using a webinar format today. So the chat function is disabled. So when you have a question, feel free to use the Q&A box because we would certainly love to hear what you have to say. We're going to start by hearing from Dr. Jamie Strange. Dr. Jamie Strange has studied bee health and genetics for over 20 years. The research focus of his lab is to understand how pests, parasites, and pathogens impact bee populations and how population genetic tools can be applied to study changes to bee populations. So we're going to hear from Jamie as to how his lab approaches these big questions and what we're going to hear a little bit about today. Welcome, Jamie.
Hi, I'm Dr. Jamie Strange. I'm the chair of the Department of Entomology, and we're here today, this morning on Waterman Farm, and we're at the Rothenbuehler Honeybee Research Lab. This lab was originally built as a facility to study honeybees and look at honeybee health and management issues. We've over the years it's evolved. A number of different research projects have been housed here, and now currently we have both honeybee research going on, and then we also have research in my lab and a couple other labs that's focusing on bumblebees and bumblebee health and genetics. In addition to what the people are going to meet today, there's a couple of projects involving bumblebees that are happening here. Dr. Mary Gardner has graduate students who are working on the effects of urbanization and urban chemicals like heavy metals associated with urban sites and how those impact bee health. And Dr. Francis Sivikoff in EEOB is also as a student working out here, looking at the impacts of fungicides that are commonly used in the environment on bumblebee health. My lab, you're going to meet three people in my lab today besides me, and they are two grad students and a postdoctoral researcher. The two grad students are Liam Whiteman and Ileana Moore, and they're going to show you and talk to you about their research projects. Basically, the overview of my lab and what we do is really to try to look at how various factors impact bee health. And so, as you probably have heard, bees, specifically bumblebees, are in some danger in the United States. We have one species which is native to Ohio, the rusty patch bumblebee, which is federally listed as endangered. We do a lot of studies on other species of bumblebees as well. There's about 28 in the lower 48 states. And those 28 species have various challenges, and we can use them, some of them as proxies to study the impacts on the endangered species. So you may see some bumblebees out, which are very common. In fact, the common eastern bumblebees all over Ohio, we see it all over the place. And we use that for a lot of our studies. Because we can get an idea of what's generally happening to bumblebees. And then we can apply some of those lessons we learned to the endangered species and things like that. So really interested in conservation, interested in how landscapes, and right now, a lot of our research is focused on urban landscapes and how those impact bee health, looking at the various pathogens. Liam's going to talk about that. Ileana is going to tell you about how those urban landscapes impact genetics and gene flow of bumblebees and the work she's doing on that around Columbus. And also, you're going to hear from Kayla Perry, who's the postdoctoral researcher in my lab. And she is really interested in how urban landscapes impact the traits that bees, how the community of bees come together on those landscapes and how they differ from urban to rural and across that gradient. So you'll hear from all of them. And what we try to do is get at these various aspects of what everybody does and bring that together then to get a whole picture on what's impacting bee health. And so that's what we're doing. I hope you enjoy the visit, and you'll then get to see the folks up in Worcester and get to talk to them. So have a good visit, and I hope we I hope you learned something today. So.
Jamie, thank you so much. I have to say I am obsessed with bees. So if you guys don't know me, I'm super excited about this virtual tour in particular. So again, thank you, Jamie, for that introduction. And as he mentioned, so current projects in his lab include understanding the effects of landscape on bumblebee pathogen and parasite community. And the impacts of urbanization on population diversity. So first, we're going to hear from Ileana Moore, a master's student, talk about some of this work. Ileana graduated from New College of Florida in 2019 with a BA in biology. She conducted internships and research projects in several areas of entomology throughout her undergrad, but she became interested in urban ecology and wildlife corridors through an internship in Latvia and a course on the subject. Her participation in the New College Pollinator Ecology Club and its efforts to conserve and educate people about native pollinators solidified her interest in pollinator conservation. So we're going to hear a little bit from Ileana about her work. And Ileana, I'd love for you to take it from here.
Hey, thank you for having me. I'm Ileana Moore, and my research focuses on the effect of urbanization on bumblebee genetics. So bumblebees have a pretty complex relationship with cities. On the one hand, they often live abundantly in urban areas and get a lot of benefit from the flowers growing in people's gardens or flowers by the sides of roads, for example. But on the other hand, cities can tend to contain a lot of impervious ground surfaces like concrete, roads, and buildings, which are areas where there's no plants growing that the bees can use, and also no soil for them to nest in. So those areas can be pretty useless to bumblebees. And because cities tend to have a high concentration of those types of surfaces, they could potentially block movement of bumblebees between different urban populations, which could potentially lead to those populations becoming isolated and becoming inbred over time as a result. So the purpose of my research, which is funded by a USDA NIFA grant given to Dr. Jamie Strange, is to figure out whether or not that's actually an existing problem for these bumblebees, or whether they're able to overcome those barriers and avoid becoming genetically isolated. So in order to accomplish that, I am sampling bumblebees from various sites across Columbus, Ohio, and taking small pieces of their legs in order to sample their DNA. And then I use a procedure called PCR to analyze that DNA. So basically how that works is I take my sample of my bee and take a little piece of its leg. Normally we do that with live bees in the field and then release them. And then we put the leg pieces in this object, which is called a well plate. So here you can see all these little legs. So these are all from different individual bees where we're getting the DNA from. And then within each of these individual wells, I'm conducting chemical reactions that allow me to replicate and amplify that DNA in order to analyze it. That also involves the use of this machine, which is called a thermocycler, that takes the reaction through a series of different temperatures that allows the reaction to actually go to completion after I've added those reagents. At the end of this, after I've amplified that DNA, I'm able to analyze different specific loci within each individual bee, and then figure out how closely related individual bees are across these different populations. And from there, I can find out whether or not dispersal and interbreeding is actually occurring between those different populations. And from there, whether the amount of impervious surface in the landscape is having any effect on that dispersal and interbreeding. So ultimately, this research could give us some good information about how urban bees are going to fare in the long term, whether we should be concerned about them, whether we might want to alter city planning, city landscapes in some way to make it easier for the bees to move across these landscapes. And it could also provide some context on the potential of cities for conservation, because we tend to think of conservation as something that happens not in the places where people live, but out in distant natural reserves and places like that. But that's not really realistic because, again, pollinators do live in cities a lot, and we depend on them for the pollination of urban gardens, suburban gardens, parks, places like that. So it would be beneficial not only to the pollinators that need help with their conservation, but also to people that live in cities to focus more on urban ecology with research, which is part of the reason I'm focusing on this.
Liana, that is so cool. I just love, well, particularly that you're sticking little bee legs into wells and finding out so much information by using that methodology through PCR and then thermal cycling. So thank you so much for giving us a little glimpse into what it is that you're doing to understand how urbanization is impacting native bees and et cetera. So thank you so much. And we're going to actually learn a little bit more about this project from Liam's perspective, who is looking at basically parasitization under certain conditions. So Liam is going to tell us about the parasite side of this work. By understanding how often bees are parasitized under certain conditions, we can better determine these relationships and then understand the health of the bee in this particular community. Liam received his BS in entomology with a minor in GIS from Oklahoma State University. So welcome, Liam.
Hey, everybody. So my name is Liam Whiteman. I'm a graduate student with Dr. Jamie Strange, and together we are studying the pathogens and parasites that make bumblebees sick, or infect bumblebees. And we're studying these organisms across different environmental gradients, such as urban to rural, or even high elevation to low elevation. The reason why we're focusing on pathogens and parasites of bumblebees is because we know that some bumblebee species are getting harder to find. We know that habitat loss and disturbance play a big role in that, but we also know that pathogens and parasites spill over from domestic bee colonies into wild populations. And we're really interested to know how these different environmental habitat structures impact the prevalence of these pathogens.
Very cool. And so when you're asking these particular questions, what sort of methodologies are you using in order to find pathogens, find parasites on bees?
Yeah, so the first thing we do is we go out to the field and we collect the bumblebees, we ID them, we bring them back to the lab, and then we immediately start dissecting them. I've got Cam here dissecting bumblebees, and what we'll do is we'll pin the bee, open up her abdomen, collect any macroparasites that may be around, and then we preserve the guts. Later, we'll take the guts, we'll do a DNA extraction, and we'll run PCR to amplify any DNA that might belong to, a disease or pathogen of the bumblebee, see if those are present.
We're showing right now the inside of what you might find when it comes to dissecting a bee. We're looking, so what sorts of things can you find? Give a couple examples. I know we're looking at a fly, a maggot actually, that's attached to an air sac right now. Is that a common occurrence, or what else might you locate inside of a bee?
Yeah, so the conopid flies are, I wouldn't say they're necessarily common, but they're part of the bee, the bee's environment. These conopid flies parasitize a bee. The female will lay a single egg, and inside of the bee, that egg will hatch and grow into a maggot, much like you would see, in like rotting garbage, essentially, but inside of the bee. So you're seeing the conopid fly. Other things that we find Crithidia, Nosema. We can find horsehair worms, which are really interesting and a unique thing to find inside of bees. Yeah, there's all sorts of things that live inside of a bumblebee.
That's crazy. Well, you mentioned Crithidia and so what are those exactly? Are those worms? Are those, what do those look like?
Yeah, so that's a really great question. Those are different like microorganisms. So Nosema, I guess the new term is Veromorpha. It's a fungal parasite that lives inside of the bee, and it can cause all sorts of negative impacts on the colony's health, their ability to collect pollen, their ability to collect nectar, their ability to reproduce. So these are all different things that impact the overall health of bumblebee colonies. Yeah.
Awesome. And so when you're conducting this work, what question are you hoping to have answered? So looking across the urban landscapes, what information are you hoping to be able to answer or provide to others when looking at the pathogen load or the parasite load over time?
Yeah, so great question. Ideally, we would like to be able to look at an environment or an ecosystem and make some sort of assumption as to what we might find in that environment pertaining to bumblebees. So in an urban setting, do we find more pathogens and parasites in urban settings? That's important to know when we talk about bumblebees like the rusty patch bumblebee, which is a federally listed bumblebee. So we want to be able to make some sort of assumptions on environments and ecosystems based on what we gather from this data.
Awesome. Thank you so much, Liam. I really appreciate your time and showing us some of the gory details of what it is that you have to do in order to answer some of these really important questions about the effect of urbanization, or basically growing cities, and what that might mean for bumblebees. So thank you.
Absolutely.
Oh, thanks. Yeah, and now we're actually going to switch over to our Worcester campus, and we are going to learn a little bit about Kyla Perry's work. She's a postdoctoral scholar in the Strange Lab. She received her BS in biology from Baldwin Wallace College and her PhD in entomology right here from OSU. Her research interests are in biodiversity and conservation of insects, insect ecology, and invasive species. And her area of specialization, I would say, is really forest entomology, but I don't want to break that down in such simple terms for Kyla, because she has so much that she's able to really do within entomology. So I'll let her tell a little bit about her work in functional traits, which she will be able to describe. And welcome, Kyla.
Hi, everyone. Thanks so much for joining us today. As Kim said, my name is Kayla Perry. I'm a postdoc working with Jamie Strange. And my research interests in this lab are understanding how major landscape level change, such as agricultural intensification and urbanization, impact bumblebee communities. And so the project that I'm working on currently is in collaboration with Dr. Claudio Graton at the University of Wisconsin. And so Claudio's team went out into the field. They sampled bumblebee communities along an urban-rural gradient surrounding Madison, Wisconsin, and then they sent me the bees, which is where I come in with my research and my research questions. And so once I get those bees, I pin the specimens and I identify them. And so I am looking at bumblebee communities and trying to understand how major landscape level change impacts bumblebee communities using a couple of measures. So of course, we can look at species diversity. We can look at what species are found at these different sites along this urban-rural gradient, and we can sort of understand who's there and who's missing. But as Kim mentioned, I'm also really interested in understanding functional traits of bumblebees. And so this is looking more broadly, not just at species, but looking at their traits and tying those traits to functions. So if you can come over here, you can see some examples of what functional traits are and what I'm measuring. And so one example is looking at wing length and wing width. So we can measure the length of bumblebee wings, which gives us some information about their dispersal capacity. I'm also measuring traits on the bumblebee head. And so we can look at head width, we can measure antennae length, we can look at how hairy the bee is as well. And these give some information on foraging capacity and carrying pollen in the environment. More on pollen, I'm also measuring bumblebee legs and some traits surrounding the pollen basket. So this here is the bumblebee tibia, also known as the pollen basket. And you can see that there's also a bunch of hairs or setae that are coming off of this pollen basket. And so I'm measuring the length of the pollen basket and also measuring the length of these hairs. And so here is sort of an example of what that looks like on a bee that has a bunch of pollen in their pollen basket. So we can see that the length of the tibia is going to impact how much pollen they can carry, and also these hairs here sort of attached to the pollen. And so that can also affect the amount that they're able to carry in the environment. And so we can take this trait information and understand how landscapes are impacting bumblebee communities by seeing if traits are over or underrepresented in certain landscapes, and whether this is happening sort of similarly in ag-dominated landscapes versus urban-dominated landscapes. And in particular, I'm really interested in some of the sites sort of in the middle of this gradient where you have less ag and less urban and more natural habitat. And surrounding Madison, Wisconsin, we actually have a lot of forest habitat and wetland habitat. And so what does this mean for bumblebee communities if we see more natural habitat in the landscape? Does this affect the traits or the species that we find? And what does this mean for natural habitat conservation and the health and protection of bumblebees?
Kayla, thank you so much. Yeah, and I had talked a little bit about the fact that forest ecology is one that is really an area of interest for you. And so,
yeah.
Are you starting to find anything in your work that is giving you hints as to what this research is going to find?
That's a great question. So I guess in urban and ag dominated landscapes is what I've looked at most closely so far. We're seeing that we find smaller bumblebees. We find bumblebees that have less hair on their bodies, and also bumblebees that have larger eyes. So there are some patterns that are occurring similarly in ag versus urban sites. And so right now, I'm diving into the more natural landscape features. And so we're finding that forest fragmentation and also the size of the largest contiguous patch of forest surrounding a site might be playing a role in terms of what traits we're finding and also species diversity.
That's so exciting.
Stay tuned.
I know, this is exciting. I love it when science is kind of in the middle of finding things because there's still more to learn. It's like reading a good book. And I am so sorry because I think I was saying Kyla the whole time. That just goes to show you that even on Zoom, you can get this. You know what I mean?
Not a problem. Absolutely.
So it's Kayla, everyone. That was completely my fault. And like I said, even nerves can hit you as a facilitator and something that you love so very much. So Kayla, thank you so much for telling us about your work. And we're actually going to transition over to Dr. Reed Johnson. So I want to give a huge thank you to everyone that presented from Dr. Strange's lab. Thank you for showing us a little bit about bumblebees and again, why they are so important, what kind of questions we're asking, and why they are relevant to people who may not always think about bumblebees and native bees. And we're going to hop over to Dr. Reed Johnson's work to learn a little bit about honeybees now. Dr. Johnson's program focuses mainly on the understanding the relationship between pollinators and their environment, with an emphasis on pesticides and understanding the physiological basis of pesticide toxicity. And again, he focuses primarily on honeybees, but he's going to tell us a little bit about what that means today. So welcome, Dr. Johnson.
Thanks, and welcome to the Bee Lab here in Worcester, where I'm based. This building was originally built for a beekeeper education program, and you could actually get an associate's degree in beekeeping from ATI up here. But that program closed long ago, and so now it's really devoted to research, which I do here primarily on honeybees. And as you mentioned, my real interest is in pesticides and how pesticides affect honeybees. And we do that in a couple different contexts, as well as how bees interact with the environment. So Hannah McKenzie, who's going to be next, and Chia Lin, who's down in Columbus, are going to tell you some about the interaction between honeybees and soybeans and the really underappreciated potential interaction there. And there's certainly cause for concern about insecticide applications in soybeans during bloom. It's quite possible that an insecticide applied during bloom could kill the bees that are out there in that soybean field, potentially increasing the soybean yield and certainly harming the honeybees and the beekeeper who manages the colony that they came from. But a lot of my pesticide work is actually in almonds, and obviously we don't grow a lot of almonds here in Ohio, but out in California, they grow really a majority of the almonds produced in the world, and they require honeybees for pollinating that crop. About 80% of the nation's honeybees go out to almonds in California each February, including beekeepers from here in Ohio will transport bees out there to service the almond pollination need. And the problem with almonds is that they apply pesticides when those trees are in bloom and the bees are pollinating the flowers to produce the almond nut. And so what I do is I test the interactive effects of the different pesticides that are applied in a tank mix, because often pesticides are not applied alone. You may have an insecticide and a fungicide and a spray adjuvant all together in the same tank, and that mixture is applied together. Now, we know a lot about the toxicity of each of those components, the fungicide or the insecticide, but we don't know what happens when they're mixed together. And out in almonds, because there's so many bees present, obviously they're not going to be using an insecticide that's highly toxic to bees. But what happens if that bee-safe insecticide becomes more toxic when it's combined with a fungicide and a spray adjuvant together? So that's the kind of testing that we're doing here. And to do that kind of testing, we go out to our apiary that's just outside this building, and we get bees, a frame of developing bees from one of those colonies. And so we've got a frame of day-old bees. And we just harvest these young adult bees off of a frame like this every day so that we can have a uniform age cohort. And then we put them into our high-tech ice cream cups here, which are our disposable cages for treating the bees. And then, so these bees that are in this particular cup would be sprayed with a combination of pesticides, an insecticide plus a fungicide plus an adjuvant, or possibly just one of those compounds. We try to do a full mixture, a full suite of different mixtures. And we would apply that at the field rate and at multiples of the field rate in order to create a dose response curve, which is the reason we have a number of these bee cups here. And so you would have one cup that gets a low rate and moving up to higher and higher rates of that particular combination. And what you'd expect to see is that as you increase the concentration, you have more and more bees dying. And so this seems like a pretty simple approach, and it is, but we can, using the results from this, we can determine which pesticide combinations cause synergistic toxicity, greater than expected toxicity to the bees, and then can advise the almond growers not to mix those particular pesticides together during almond bloom when the honeybees are out there pollinating their crop.
Reed, you mentioned a lot about almonds. How does that translate to the folks back here in Ohio?
Well, certainly we eat almonds. Actually, the large commercial beekeepers here in Ohio send their bees to almonds. So the bees that we have, they're making the honey that you might be buying at your farmer's market. Those bees could very well have spent the winter out in California pollinating almonds. So it's very relevant here in Ohio. Also, some of these same pesticides that are used in almonds are also used in crops that are grown here in Ohio. And the tank mix combinations that we find could be harmful in almonds may also be harmful in the cucurbit crops, pickle pollination or pumpkin pollination, and the bees that are brought in to pollinate those crops as well.
Sure, that makes a lot of sense. And I wanted to pause for a moment because I know I've been asking a lot of questions, mainly because, of course, as I mentioned, I love bees. And so I wanted to pause for a moment to see if anybody who's viewing right now has a question for Dr. Johnson about any of the work that he's talked about. Looks like we don't have anyone yet. I know I'm fascinated. And so can you talk a little bit more, Reed, about this combination of pesticide, basically the different chemicals that are put together to form pesticides? You had talked about how the combination of the toxicity is a big piece. What are those different things that can be combined? Can you go into a little bit more detail there?
So yeah, it's quite common to make one application in the field and it has multiple constituents in the tank. Often, almost certainly, there's a fungicide involved in that spray application. There may also be an insecticide. And then the third component is often a spray adjuvant, which is intended to improve the penetration or the spreading capability or possibly the sticking of that pesticide application, kind of to modify the physical properties of that pesticide when it goes on. And so you have at least these three different things: insecticide, fungicide, and adjuvant. And it's what we found in doing these relatively simple assays is that particular insecticides interact with particular fungicides, and that interaction is made worse in the presence of certain adjuvants. So you really can get particular killer combinations that could actually kill bees in the field. Meanwhile, there's other combinations that appear to be relatively safe for bees. So we're just trying to identify which combinations pose the greatest risk.
That makes a lot of sense. Now, I've heard rumors that there is something called, I think, the powder tower or something of that nature that's in the Worcester campus. Can you talk maybe a little bit more about that?
So the Potter Tower is this device that was developed in England in the 1960s. And it's essentially a sprayer on this about 1 yard tall tower. And what it does is you can actually fill this apparatus with your tank mix combination as it would be applied in the field and apply it to just the size of one of these cups. So rather than spraying a whole field, you're spraying just the area of this cup. And this allows us to relate the label application rate. Rather than spraying a whole acre, we're just spraying the area of this cup, and we can do the calculations so that this cup is getting exactly what these bees would get if they were out in the field and exposed to the label application rate for these different pesticides.
That makes a lot of sense.
It's a good simulation of field exposure in this really elegant small system that you can do inside a lab. It's quite old, but it still works really well.
No, that's a great way of doing it. Like you said, so that it's not over this giant field, you can actually really control it. Well, and that brought up a question actually from Matthew. Are residuals from pesticides also a concern, or mainly just products being applied with honeybees present, like direct contact?
So it depends on the particular product. EPA maintains a database of the residual toxicity to bees for a number of different products. That information is also usually listed on the label, how long a particular application may be toxic to bees. Many of the newer chemistries break down relatively rapidly in sunlight. So the residual toxicity is less of a concern now than it was with older chemistries that are being phased out. But residual toxicity is definitely an issue.
Sure. Well, and we have one last question for you. So the question's about climate change and how it might be affecting pesticide application and pollination. So like the wildfires in California and the almond crops out there, how is all of that playing together? And are you starting to look into questions related to climate change and pesticides?
So climate change and honeybees are a challenging area to grapple with, because ironically, honeybees do better in the tropics. So here in Ohio, the honeybees maybe winter kills a lot of bees. So warmer weather is generally better for honeybees. That being said, I think if warmer temperatures and climate change cause a need for increased pesticide usage, obviously that's going to be a bad thing for bees. And particularly related to the fires and the drought out west, what that's likely to do really has less to do with the bees and more to do with the ability to grow almonds out there, and that they may not have the water in order to support this vast over 1 million acres of almonds that they've got. And if they can't grow almonds because they don't have water, then they don't need bees either, which is going to harm a lot of beekeepers. They make a lot of money pollinating almonds, and I suspect there'd be a number of beekeepers that go out of business if that wasn't a regular income stream for them.
Sure. So sad all around, except yeah. So really complex answer to that question. Actually, it's going to be interesting to see how things move forward.
It's a complex issue. Yeah.
Mm-hmm. Yeah. Well, thank you so much, Reed. Thank you for giving us a breakdown of what it is that you're looking at right now. I know that you have so many other research questions that you're tackling as well, but at least giving us a little bit of an insight as to some of the bigger pieces of work that you're doing. I really appreciate that. And I know that we're going to move on and talk to Hannah as well and learn a little bit about her work. So Hannah McKenzie will be taking over to talk next about actually one of my favorite behaviors about honeybees, which is the waggle dance. Hannah has a BS in entomology and horticulture from Ohio State University. And so she is going to actually show us some behavioral traits related to soybeans here. And Hannah, welcome, and talk a little bit about the work that you are doing and show us the setup that you have going on.
Hi, so thanks for that introduction. So I'm Hannah, and I'm really excited to have the opportunity today to talk about my research with honeybees and soybeans. So there's a lot that we can learn from observing honeybee behavior. And one of the ways that we can do that is by using something called an observation hive. So this here is one such observation hive, and it's basically just a small colony of bees with glass walls. So we can easily look inside and observe the bees going about their day-to-day activities without actually having to open up a whole colony and disturb the bees. Now, this wall here is an external wall next to the colony, and there's a tube here that connects the hive to the outside world. And you can actually see there are bees moving in and out of the colony, collecting resources and bringing them back to the other bees. Now, one of the things that we can observe using an observation colony such as this one is something called the waggle dance. So bees actually have a way of communicating with each other called the waggle dance. And what will happen is a bee will go outside to forage for resources. And if she finds a really good resource, she can come back to the colony and inform the other bees of where that resource is using the waggle dance. So what she'll do is she'll shake her body really quickly and move in a certain direction on the comb. And the direction that she moves on the comb tells the other bees what direction they need to fly compared to the position of the sun in the sky to find that resource. The duration of her dance tells the other bees how far they need to fly in that direction. So that combined duration and directionality tells the other bees how to get to good resources to collect for the colony. Now, one of the things that is really cool about this waggle dance is that we as humans can actually interpret it ourselves. Now, bees can do it naturally. It comes very naturally to them, but we have to work a little bit harder to interpret where the bees are dancing for. So something that we do is we can open up this bottom panel of the observation colony and film the bees dancing on the comb with a camera. Then we can go back and look at that footage, and we can trace the dances using a software and then map those dances onto an actual map to see where the bees are dancing for and what resources they find attractive. One of the things that we're applying that footage and tracing of waggle dances to is determining when bees are foraging in soybean flowers. So what we can do is we can look and see on what days bees are particularly attracted to soybeans, and then we can use that with some other research we're doing to show when soybeans are most attractive to honeybee foragers. This information could be used to help us inform soybean growers when there are bees pollinating their fields, and this can help them to not expose those bees to unnecessary pesticide exposure. So these observation colonies are really good both for just general bee behavior observation and also for measuring things such as the waggle dance. I'd be happy to take any questions about the observation colonies or waggle dances or our research with honeybees and soybeans.
Hannah, if I remember correctly, there is no pollination required for soybeans. Is that correct?
Yes, soybeans are a self-pollinating plant, which means that they don't need an external pollinator in order to produce soybeans. However, there are a lot of studies that show that when you do have pollinator activity in soybeans, it does significantly increase the yield of those soybeans. So it is in soybean growers' best interests to support pollinator activity in their soybeans so that they can have greater yields.
So then did you mention, or did you talk about it all, like, are there soybeans right outside that wall that's connected to this observational colony, or how does that all work out?
So there are soybeans in this general area, and because of that, we don't actually use this specific observation colony for measuring waggle dances for soybeans. It's important that the resources we're trying to look for dances for are actually at least maybe half a kilometer away from the observation hive. Because if the resources are like right outside, the bees won't perform a very good dance, and it's really difficult to actually measure that. So we've actually set up some other observation colonies at an external location that are at least half a kilometer away from the nearest soybean field. And we're using those to track waggle dances in soybeans.
Very cool. What are these bees interested in?
Well, we have a lot of things around here. There's a lot of pollinator habitat around here that has a lot of wildflowers. There are soybeans around here that they could very well be pollinating. So those are probably the main crops around here that they're interested in at the moment.
Very cool. Any other questions from the audience? Well, it doesn't look like it right now. Hannah, thank you so much for describing your work. I think, like I said, waggle dances are adorable, and it's just such a neat form of communication for this particular insect to be able to provide to the rest of the colony. So thank you for sharing a little bit more about that. And we are actually going to now shift back to Columbus campus to learn a little bit more about what's going on in Dr. Reed's lab from Chia Lin. She is a research scientist in Dr. Johnson's lab. She works primarily in the Columbus campus, but will be telling us about a few projects she is working on. And she's going to start off in the field, talking to us a little bit about some similar work as Hannah, talking to us about the relationship between honeybees and soybeans. So we are going to take it away to Chia.
Hi, I'm Chia, and welcome to my laboratory here at Waterman. We have these cages that we're doing. I'll explain a little more on the experiments we're doing with the soybean cages. But you know, soybean is probably one of the most cultivated crop in the United States, with corn. They're top number 1 or 2 back to back to each other. And in the Midwest, especially, we got a large acreage, a huge amount of land here is covered in soybean crops. And as we discussed before, soybean, because it does not require pollinators to produce the crop, the interaction between soybeans and honeybees, or bees in general, has always been overlooked. But in our lab, we actually spent quite a few years looking at honey samples collected from all over Ohio. And we found there are soybean pollen in our honey samples, especially from regions where soybeans were predominantly. That soybean has a potential to provide some food to honeybees as well as other pollinators in this landscape. So now it got us to think, you know, traditionally, those large monoculture corn soybean landscapes has been viewed as a very low quality for honeybees because it's not perceived as a good foraging habitat for bees. And also with regard to the pesticide use to protect those crops, it has the potential to harm the honeybees. However, what about if we changed our thought a little bit? And what about if we could turn these large acreages of soybean fields into food for honeybees? So they got us to our experiment here. We're interested in finding the soybean varieties that could produce the most amount of nectar to support the bees. There are hundreds of soybean varieties on the market in Ohio, in our region alone. And every year we get a lot of new soybean varieties coming up. So we did a lot of surveys. We have actually in the past 2 years surveyed over 150 soybean varieties, and we measure the flower size. We measure the amount of nectar the flower produce and the amount of nectar, sugar concentration, and we counted the bees were visiting in those varieties and determine which varieties are most attractive to honeybees. And here in our cages, we have a total of 12, and each cage contain 8 varieties. So actually there are 2 sets of experiment here. We have a total of 16 soybean varieties. And these are varieties either that we have determined from our previous survey, they are nectar rich and attractive to bees, or some of the varieties were donated by farmers. They're interested to know how well their soybeans are doing in this kind of setup. So let's come into one of the cages. I'll show you what's going on here. So here is one of the cages, one of the 6 cages that have a honeybee colony here. I'll walk toward to them. It is about to rain this afternoon, or actually anytime now. So the bees are not super active, but you can see they're kind of hollering. This is the same hive that the same kind of setup that Ree was showing in Worcester earlier. And in this cage here, we have 8 different varieties. They're like little different in little sections. And you can see some of the varieties. We have purple and white flowers for soybeans. And I'm hoping some of the bees will forage for me, but they, you know, when the rain is in the forecast, all the bees, all they want to do is to go back to their hive and stay dry. So we'll see. I'll point one out if I see one. But with this experiment here, so we are comparing how the soybeans do with and without honeybee, the presence of honeybees in the cage. Obviously, in the cages that without a honeybee colony, we're looking at the yield in the absence when the pollinator is not present. How much yield does that produce? Does the different varieties produce compared to in the cages where the honeybee foraging activities are present? And another thing that we have, so besides in addition to comparing yield, we're also looking at the because we exclude the bees from the soybeans flowers, so we have full control of the flowers, so we could actually measure the total potential amount of nectar production in the soybean varieties, and we can come up with a rank of the most nectar rich varieties. And our next step of the experiment would be going into like actually plant this selected varieties in the field and then see how they do. And we can calculate at a landscape level how much nectar do the soybeans supply, how much nectar do the soybeans supply to the pollinators. And some of them start to fly out. So this actually brought us back to the concept of pesticides. Now we could provide more food by growing the nectar-rich varieties, but we still cannot steer away from the impact of pesticides. And in our lab, as Reed has discussed earlier, we're doing a lot of toxicology experiments, bioassays, and so in Worcester, even though right now our project, the pesticide bioassays are mostly focusing on almond, but we were hoping in the future we'll be able to expand to more crops. So we have in Worcester, most of the focus was on the spray simulation on adult worker bees, but there are other cases, there are other components of the beehives. So for example, each colony would be has this very essential role called the queen. And this is the queen, the sole individual in this colony that produce more worker bees that sustain the colony. So in Columbus, we're doing this, the pesticide bioassay as well with the queen. Essentially, we use this device called a swarm box. It's a wooden box sealed up with screens for ventilation, and we provide this box. It contains nurse bees. It contains food, pollen, and sugar nectar, and we give them the larvae of very young bees. And in this setup, the worker bees would consume the food and in the process to produce to make queen cells that would be the production of the queens for the colony. So with when we feed those colonies contaminated pollen contaminated with pesticides, we're able to evaluate how does this pesticide affect the development and survival. And also with this method, we could harvest the royal jelly that the workers produce to feed the queen larvae. And we could actually measure how much of those active ingredients in those pesticides are transferred into the royal jelly, which is the food that the queen larvae and also worker larvae consumed in their hive. Now, speaking of worker larvae, another very cool component we're doing in the Columbus lab is we also do artificial larval rearing in the laboratory. And this is the fascinating techniques that someone has developed. We could transfer very young worker bee larvae into the cell culture plate. And then we feed those larvae with royal jelly infused with pesticide compounds or combination of pesticide products. And we could track those development of those larval worker bees through adulthood and monitor the survival, and it allows us to determine whether the pesticide compound by itself or a combination of the compound could have a lethal impact on the development of the bees.
This was wonderful, Gia, and we were showing some of the photos that you provided to show kind of how things go when you are rearing both queens and also those larval bees and what that might look like at each stage. So thank you for providing those, because it really does show kind of what things really look like behind the scenes. So I appreciated that. Any questions? Well, Gia, I have one. So when you're looking across the different varieties of soybeans, are you starting to notice anything?
Yes. So I forgot to mention, we look at, we look at the color, sugar concentration, and nectar, and the amount of nectar that the flower produce. What we discover is the color of the flower doesn't really make a big difference. What the bees care most about is the amount of nectar the flowers produce. So the varieties that are able to produce more nectar are generally more attractive to bees. And you can actually kind of tell that some of the beans here are taller than others. So the different varieties also have a slightly different growth form. And that's kind of interesting. We would look into whether or not the height, the growth form, and how the architecture of the plant influence the attractiveness of the bees. And that part is ongoing, that we haven't really have a a result yet.
That makes a lot of sense. But, and I was wondering too, when you mentioned yield with regard to this relationship between honeybees and the different varieties, do you mean that they simply produce more of the same, or are you noticing anything different about the type of yield that's produced from those soybeans?
Yeah, when the honeybee is present, in our cages, I think one limitation with the cage experiments, we kind of block out some of the sunlight, so the plants are not able to photosynthesize in their full potential. However, we did find that plants in the cages with honeybees produce more full pods. So soybean can produce up to usually 3, but occasionally 4 pods, because that's how many ovules are in each of the flowers. And we found more of the pods, there are 3 or 4 seeds in the pot in the cage when the honeybee is present, compared to the cages where the plants are not pollinated by honeybees. But at this stage, we haven't noticed a significant increase in the total yield. That's the total weight of the seed produced by a plant. Although there are some studies by other people in the field that have found it could be up to 15%. It was one study saying 50%, but somewhere in that range, it could definitely significantly increase the yield in some circumstances.
That's neat. Yeah. So at least some sort of improvement is what it seems like researchers are noting. No, that's fascinating. Thank you so much, Tia. It was so nice to see this. Yeah, I appreciate your time, especially because you are definitely trying to outrun the rain. So thanks for taking the risk for us.
Yeah, thank you. The bees are starting to flying a little bit. I haven't noticed any foraging yet. This is another thing, actually. Sorry, I just, when the weather is warmer, the flowers produce more nectar. There's one. So they're more likely the bees are more likely to forage on the flower on a hot day, but obviously it's not hot enough right now.
That's so neat.
Thank you.
Yeah, thanks, Gia. Perfect. Well, thank you all for this wonderful brief kind of introduction into your work. I know it's only a small piece of what you guys go through day to day, month to month. So it was such a joy to learn about every piece of it. I do want to give some additional time for questions, but before I do that, I wanted to quickly thank Shiftology for their assistance in all of this. They're kind of behind the scenes helping us orchestrate all of these virtual tours. And then at the end, when you do close out from this, you'll see a short survey pop up on your screen, as we'd love to hear your feedback to know what it is we can improve, what you enjoyed, and also if we should do more of these. At the very least, I hope everyone stays dry today. I know there is supposed to be some pop-up showers, at least in the Columbus area. So for those of you in the rest of the state, I do hope that you are able to stay safe and dry and enjoy some of these hot, sticky Columbus or Ohio temperatures. And thank you so much for joining us today. I appreciate your time. We look forward to hanging out with you again soon.